Welcome everyone to today's webinar, which is about understanding the applicability of COVID-19 vaccine trial results to low and middle income country settings. This webinar is co-organized by the COVID-19 Clinical Research Coalition and the Moru Tropical Health Network. Today's webinar is going to be recorded and it'll be posted online later today on the Coalition and the Moru websites. Now, the COVID-19 Research Coalition was put together early on in the pandemic in order to facilitate and fast-track clinical research in low and middle-income countries. It's composed of multiple working groups, and I'm the co-chair of the Study Design and Analytics Working Group for the Coalition. And one of the reasons to have this webinar today is to advertise this working group and our services. So apart from a few exceptions, almost all clinical research that's reported so far in COVID-19 has lacked coordination. And as everyone knows, some small underpowered studies have had inordinate influence on clinical practice. And the goal of this Study Design and Analytics Working Group is to provide advice about optimal trial design and data analysis. So please use our services, please get in touch and you'll find the email address to reach us in the chat. So I'm delighted today that we have 3 world experts on vaccine trial design and implementation. And their expertise ranges from the very, very technical to the very practical. And there's going to be a 15-minute Q&A session after the talks. And I'd like to encourage everyone to post your questions in the Q&A, and also to upvote questions that you think are good by clicking the thumbs up icon. And I'm going to read through by order of popularity. Now, the first talk today is going to be given by Dr. Rebecca Khan, who is a postdoctoral research fellow in the Center for Communicable Disease Dynamics at the Harvard Chan School of Public Health. And in 2018, she published a paper in the American Journal of Epidemiology, which was about how to analyze vaccine trials in epidemics with mild and asymptomatic infections. So she was 2 years ahead of everyone else in thinking about how vaccine trials should be designed for COVID-19. And she's the best person to kick off this webinar. Rebecca, please, over to you.
Great. Thank you so much, James, for that introduction. And thank you so much to the organizers for inviting me to participate. So as James mentioned, I will be talking about some work I've done on designing and interpreting vaccine trials for epidemics with mild and asymptomatic infections. So first, I want to take a step back and look at the landscape of COVID vaccine trials that are currently ongoing. So vaccine candidates are being developed at an unprecedented speed. This is a screenshot I took of the New York Times coronavirus vaccine tracker last week. And you can see that there are dozens of vaccine trials that are ongoing. The scientific progress this year has been truly remarkable. Now, most of these trials, as you can see, are in phase 1 and phase 2. So these are the early studies that are in smaller groups of people that aim to assess the vaccine candidate's safety and immunogenicity. Now, 11 of these trials are in phase 3, which are the larger scale trials that aim to assess efficacy in a large group of people. And so the design and analysis of phase 3 trials is going to be the focus of the rest of this talk. So when we think about designing phase 3 trials of vaccine candidates in the midst of emerging epidemics of emerging infectious diseases, there are a number of choices that need to be made. And in the paper I've cited below, that's in PLOS Medicine, we talk about 4 of these choices in detail. And briefly, these 4 choices include the randomization unit. Are individuals randomized to receive the vaccine or another intervention, or are clusters, such as communities or different groups of people, randomized to receive the vaccine candidate or something else? The second choice is, what is that comparator intervention? Is it a placebo? Is it another vaccine, an active control? Or is it just a delayed administration of the vaccine candidate? The third choice we discuss in this paper is the trial population. Who is enrolled in the trial? Is it a trial for the general population, or does it target high-risk groups, such as healthcare workers or other people at higher risk? And then the final choice we discuss is trial implementation. Is it trial start everywhere at once, or is it phased in over time as more resources become available, or to follow the trajectory of the epidemic? Now, many of the COVID vaccine trials that are ongoing are individually randomized placebo-controlled trials conducted in the general population. Some of the trials may use active controls such as the meningitis vaccine, and some have inclusion criteria that require people to be at high risk. But in general, this is the design of most of the studies that I've looked at. Now, when we think about designing vaccine trials, I think it's helpful to think about how the vaccines are going to be used. And in their recent paper in Science, Mark Lipsitch and Natalie Dean outline the two main ways that vaccines are used. And they use an example of protecting the vulnerable, with the example of the vulnerable being in the elderly population. So the first way a vaccine can be used is by directly protecting the vulnerable population. So on the left, you can see in this example, to protect the elderly, we would vaccinate the elderly. Another way a vaccine can be used is indirect protection. So if our goal is to protect the elderly, we vaccinate adults who are most likely to transmit or highly likely to transmit. And the goal is to break the backbone of transmission, either by keeping adults from getting infected themselves, or even if they do get infected, if the vaccine has any impact on reducing their transmissibility, this could also protect the elderly. Now, when we think about, for example, influenza vaccination, often the goal is to protect the elderly who are at risk of severe disease. But the influenza vaccines have been shown to elicit weaker responses in the elderly. So indirect protection is a key component in influenza vaccination programs. So when we think about a vaccine candidate, if our goal is direct protection of an at-risk group, this requires that the vaccine is effective in these individuals. So in this example, in the elderly, or in another example, we could think about populations with specific comorbidities. Now, in vaccine trials, it's unlikely that we will know this at the first successful interim analysis, because success will be declared when overall efficacy is found, and the trials will likely be underpowered for detecting vaccine efficacy within these subgroups. We might get some effect, but the confidence intervals will likely be wide. When we think about assessing indirect protection, this requires that the vaccine reduces transmission by preventing infection and/or reducing infectiousness. And it's unlikely we will know this for most vaccines because the primary endpoint in most trials is symptomatic PCR positive disease. So this brings us to another important choice when designing vaccine trials, which is trial endpoint. So as I mentioned, symptomatic PCR positive disease is a primary endpoint in most vaccine trials. And this is an important endpoint. However, as I just described, another endpoint, vaccine efficacy against any infection, is also important, and vaccine efficacy against severe disease or vaccine efficacy in different subgroups. So for the rest of the talk, I'm going to discuss these two other endpoints in more detail. So starting with the trial endpoint of any infection. So understanding vaccine efficacy against all infection and not just symptomatic disease is important for pathogens where people with asymptomatic or mild disease can still transmit, such as with COVID, or with pathogens where there are side effects of infection, such as congenital Zika syndrome with a Zika virus. Additionally, an unbiased estimate of impact on all infection would provide evidence about the potential of a vaccine for herd immunity. So it provides really important information when thinking about how this vaccine could be used. However, there's bias in vaccine efficacy estimation against all infection when we're considering only symptomatic infections. I'm going to walk through why this occurs. So if we think about a group of people represented by the circles here who are enrolled into a trial, and we randomize half of them to receive the vaccine and half to receive the control, we follow them up over time and we see a certain number of people get symptoms, and we can test them to see if they're PCR positive. But for the remaining people in the trial, we don't know what happened to them, whether or not they got infected. So if we take an example here where we have 10 people who receive the vaccine on the top and 10 people who receive the control intervention on the bottom, And in this example, 3 people in the vaccinated group got infected, represented by the orange, and 8 people in the control group got infected. So this means that there are 7 people still at risk of getting infected in the vaccinated group and 2 in the control group, represented by the pink here. If we look only at symptoms, in this example, only 1 of the 3 people in the vaccinated group who were infected had symptoms, and 2 in the control group had symptoms. So it looks like there are a lot more people who are perceived at risk than actually are at risk of still getting infected. And this occurs more so in the control group than in the vaccinated group. So our apparent incidence in controls is underestimated more in the vaccine group, biasing our estimates towards the null. So the key takeaway from all of this is just to say that by looking at only symptomatic infections, it's going to make the vaccine look less effective at protecting against all infection than it actually is. Our vaccine efficacy estimate against all infection will be biased towards the null. So when we think about trials where the primary endpoint is symptomatic disease, is efficacy against symptomatic disease, this is not an accurate proxy of vaccine efficacy against all infection. Now, some trials that are ongoing do have vaccine efficacy against all infection as a secondary endpoint. However, many trials do not, and that's because this can be resource intensive, because estimating vaccine efficacy against all infection can require frequent testing to identify mild and asymptomatic infection. This requires a lot of time on behalf of both people conducting the trial and participants in the trial to get tested frequently over time and not just when they exhibit symptoms. So to try to address this challenge, we developed a model of an outbreak in a vaccine trial. Now the details are described in the paper cited below that was published in the American Journal of Epidemiology. But briefly, what we did in this paper was through this model, we tested a variety of methods for estimating vaccine efficacy against all infection. And we looked at different methods that required different amounts of resources, from frequently testing everyone throughout the trial to testing just a sample of people at the end of the trial. And what we found is that we can actually get an accurate estimate of vaccine efficacy against all infection, not just symptomatic infection, by serologically testing just a sample of people once at the end of the trial, which is substantially fewer resources than we initially thought would be required. Now, thinking about some of the logistics of this, if we're serologically testing people to see if they got infected, we need a marker of infection that is distinguishable from vaccine immunity, so non-vaccine antibody. For COVID, most of the vaccines are targeting the spike protein, so for example, the N protein could be used. And additionally, for these trials, we ideally need serology at the beginning of the trial to identify if anyone has been infected previously, and then we need post-study sera from a sample or potentially all participants. So if you're interested in reading more, we describe all these details in the paper, but essentially we found that it is possible to get estimates of vaccine efficacy against all infection, which is an important endpoint with fewer resources than we thought would initially be required. Now moving on to vaccine efficacy against severe disease and vaccine efficacy in different subgroups. So if vaccine trials are stopped based on the number of people with any symptomatic disease, they may not be powered to detect efficacy against severe disease, which is a less common outcome, or efficacy in different subgroups. Like we talked about before, this could include different age groups or people with different comorbidities. And this can limit the generalizability of the results from a trial if the trial is conducted in a population with a different age structure than one we're interested in thinking about applying it to. And in their recent paper, Mark Lipsitch and Natalie Dean outline a few solutions to this problem. So one option is to set minimum enrollment targets for subgroups, or to only perform interim analyses after a certain threshold of cases in subgroups have occurred to prevent stopping the trial before it would be powered to detect effects within these subgroups. Another option is to continue placebo-controlled follow-up for longer, for example, for up to a year, or until the vaccine becomes widely available in the area of the trial. And this can help with not only getting some of these other efficacy estimates of interest, but it can also help assess the duration of protection and other factors. A fourth option is to conduct post-approval observational studies to try to get some of these additional measures. Now, of course, observational studies are always subject to confounding, but there are methods and study designs that can help overcome these issues. So in summary, the primary endpoint in most trials, including the COVID vaccine trials that are currently underway, is symptomatic PCR positive disease, which is an important endpoint. However, there are also important implications of understanding a vaccine's efficacy against all infection, against severe disease, and in different subgroups. And there are challenges in obtaining these different estimates, but solutions exist. So with that, I want to thank you all for listening, and later on, I'd be happy to take any questions.
Thank you very much, Rebecca. That was brilliant. So yeah, all the questions, please put them on the Q&A, which you'll find at the bottom of the screen, and we'll have a session at the end. So the next speaker today is Dr. Rebecca Grace. Dr. Grace is the Director of Research at Epicentre, which is an epidemiology and research satellite that was created by Médecins Sans Frontières, or MSF. And Epicentre and Dr. Grace have a lot of experience in vaccine research in low and middle income countries. And most notably, Dr. Grace led a lot of the vaccine and therapeutics research during the recent Ebola outbreaks. Over to you.
I would like to, James, but I cannot start my video.
I think you need to be authorized.
I do. Of which I clearly am not. Well, I'll tell you what. Do you want to just go to the slides? Super. As long as everybody can hear me, then that's fine. And first of all, I should thank you before I start for inviting me and asking me to participate in this. I have to accept control. So if we go back to here, and I'll just start talking and then we can skip the video. So what I wanted to talk about this morning and what James asked me to discuss was a little bit more about what are some of the practical implications of doing vaccine trials in epidemics, like the one we're currently experiencing, and in particular in low and middle income countries. And so the first thing, which is somehow, it's not somehow, but it's often forgotten, is what assumptions do we have about doing vaccine trials in epidemics? Well, the first one is that the question actually necessitates an answer in the epidemic itself, meaning that we cannot figure out the answer to this question elsewhere. And so usually that means we're looking for vaccine efficacy and in some cases effectiveness, that we're aiming to provide a possibly life-saving intervention. And I emphasize the word possibly because we're dealing with candidate vaccines here. That we're able to provide appropriate or at least best available care to people participating in a trial. And that comes from a basic tenet, which is that there should be a benefit for communities where the research is conducted, aside from possible future use of a vaccine if it is proven both safe and efficacious. And the last part is the population of potential trial participants need to themselves recognize that the trial is important to them. And that means not just the people financing the trial or the researchers themselves, but the participants. So just some background. So I work at Epicentre, which was created by Médecins Sans Frontières, and we work in epidemiology and research and training among populations who are affected by conflict or with limited to no access to care. We have extensive experience, as James mentioned, in trials and research in epidemics, and in particular among populations in crises. And so I have to say that COVID is an exception, which I'll speak about in the next slide. But we've looked principally at different Ebola Zaire vaccines in different outbreaks in different countries, and even up until today, and looking at both multicentric and single-centric trials, which look at immunogenicity of different vaccine strategies, aiming to facilitate their use in the future. And then right now we're looking at some preliminary studies aiming to set the stage for future Lassa fever vaccines. But so what's different about COVID? Well, there is to some degree a uniqueness of COVID when we're looking at trialing potential vaccines and epidemics. The first is, yes, it's definitely a pandemic, but a pandemic but. And by the but, what I mean there is the cases are not equally distributed around the planet. And as a consequence of that, where one would trial a vaccine is, of course, very different in terms of getting a potential result, and also who benefits and who would be the most likely individual to receive it if it is in fact rolled out in the future. And one of the things that is different about COVID is that unlike many other emerging infections, the burden is very high, or quite high, I should say, in high-income countries, and that's often not the case. We also have massive global media coverage. One other point, which is just for those of you of interest, is we've had many difficulties with viruses in general in the past, and we have even more so with a new one, and in particular in vaccine development. And in COVID itself, in this pandemic, we have inequities which are highlighted in a new way, in a very different way that we've seen in the past across the planet. And we also have variable and different timescales for vaccine research and development, regulatory aspects, manufacturing, allocation, and implementation. So this is a relatively unique scenario. If we look at some of the lessons learned in terms of trialing candidate vaccines in previous epidemics, there's 4 big categories of things that could be applied to trialing a COVID vaccine. The first one is that expecting the unexpected should be taken as a given. The second is the importance of keeping everybody's papers in order. More money, more problems, meaning that we often have issues with money, which I'm sure of all of you who follow the news may have seen this already. And then the last is just to use a French expression, which is savoir, savoir-faire, and savoir-être in terms of doing that. And there's some lessons we can take from that. So right now, I'll just go through the 4 of these and some examples and potential implications for trialing a COVID vaccine. So the first one, in terms of expecting the unexpected, on the left-hand side of this slide, you'll see a list of all sorts of things that can happen. And those things happen, and for those of you in the United States, I would even add election contestation can certainly occur there in high-income countries. These are examples from low and middle-income countries, but there's no reason that some of these wouldn't also occur in high-income countries. And so there, in terms of the conduct of a trial itself, the key to risk mitigation here is the presence of senior staff. And so what we have there is, what I mean by senior staff is that the study staff itself need to have both management and contextual experience. That's not something that is often discussed in terms of the conduct of trials, but it's an important aspect. The second is that risk management in and of itself of a trial should include scientific risk management. And this also speaks to the importance of ensuring that the population themselves who would potentially be enrolled in a trial also understands what the trial is about and that there is some potential benefit conferred to them. And so the question which is most important to address in the conduct of a trial is what is achieved if study failure is likely to be the outcome? What do people get if the primary outcome actually isn't successful? And ensuring that in terms of the conduct of a trial of this magnitude is extremely important from my perspective. The second is with respect to papers and keeping things in order. So one of the key difficulties that's often encountered in the conduct of trials is that roles and responsibilities of who's involved may not necessarily be as clear as they should be and are not necessarily communicated. We can see that a little bit in the general media reports about COVID vaccines, where it may not necessarily be clear always who's in charge of what and who is actually in charge of releasing which results, when, and to whom, and for what end. And sometimes those semantics can be things which add additional confusion and lead to problems later in terms of the interpretation of results and how they're communicated. The next part is, in addition to all of the bells and whistles that one needs in terms of the conduct of a trial to ensure its scientific rigor, there also needs to be what's sometimes called an operational master file, which ensures that all authorizations have been obtained, that all personnel are following local laws and have clear HR policies, including a salary scale and job profiles. There's financial accountability and benchmarking for use of funds, and that there's also some degree, and in many cases it should be a higher bar of anti-fraud, conflict of interest, and security policies, among others. The last part of this slide deals with keeping documents directly related to the study in order and addressed. So not only ERBs and NRAs, but also ensuring that adherence to international norms and standards is done, but that doesn't have to be complicated or a burden. That's sometimes used as a way to, as a tactic, if you will, to inadvertently make things sound more complicated than they are. But the heart of all of this is to ensure scientific rigor while ensuring the protection of participants. The third one deals with money. So there's lots of money going around in COVID, and sometimes more money leads to more problems. Research is expensive, and doing vaccine trials is expensive, and it often represents a disproportionately large sum compared to the needs of a population or to those providing care. And what that means is that we do have to take care in terms of how money is discussed, how it's allocated, and what it's used for. Actually, I repeated myself, but you guys get the drift. The last part is having these high budgets and with them high salaries do not necessarily translate into meaningful results. And so what that translates into something like COVID is that one can spend $1 trillion and still not wind up with a meaningful result. There's not a direct relationship between those things. And additional means, whether they're human or financial, are not always the way to solve problems in a trial. The fourth one, which is this tenet, which is savoir, savoir-faire, and savoir-être, is that the paradigms of R&D that we're looking at, which include both trial design, which Rebecca talked about in the previous talk, and implementation, need to be treated holistically. It's very difficult to do this in a vacuum. And one of the major points in terms of leading to a successful trial, particularly in a difficult context or a difficult political context, would be ensuring that all of those groups and all of those aspects are treated as a whole. In terms of low and middle income countries in and of themselves, there needs to be some degree of flexibility and adaptability in terms of the superimposition of high resource concepts, if you will, without compromising quality, meaning that there's not necessarily a one-size-fits-all solution in order to respond to international norms of clinical trial conduct and design, and we do need to be careful about how those things are done. Semantics are not universal. That's something, especially in the case of COVID, where we're dealing across countries and across contexts and with different people using different words. It's important not to assume that those things are a given. That in terms of what's given back, if you will, or the legacy of a trial itself in a community and among its staff, that those claims concerning what will be transferred in terms of capacity building and future benefits should be tempered and transparent for both participants, authorities, and study staff. This isn't that complicated. It just means being honest about what's given. And the last one is that healthy participants have different needs than those in a therapeutic trial. And so it's important to always remember that in a vaccine trial, we are dealing with individuals who are healthy. The last one, which is more about communication, and I think Cherry will talk about this in the next talk, is in terms of the importance of clearly communicating the objectives of a trial. What are the expected outcomes of the trial? Who benefits and who doesn't from the trial? And whether there are actually future use possibilities or not. All of that certainly needs to be clarified, certainly for the participants, as well as any other stakeholders involved in the trial. And that the results, irrespective of what they are, need to be made public to participants, authorities, and the scientific community. And then I'll just end on some last thoughts about COVID-19 vaccine trials in low and middle income countries. Well, the first one is that different vaccines are likely to perform and be used differently, and their status will matter. So Cherry will talk about this in her talk, but whether a vaccine is under an emergency use access license or whether it's fully licensed or it's still being trialed makes a difference. And it makes a difference in terms of how that vaccine moves forward. The second point is the importance of uncertainties and that not knowing is normal. In the example that Rebecca gave, and she touched on this, if we have a vaccine whose primary outcome is aiming to look at efficacy, vaccine efficacy among symptomatic cases or clinical efficacy. And we find that 50% vaccine efficacy is the point estimate found in the trial. So we might have 100 cases in the placebo group and 50 in the vaccinated, but the confidence interval around that estimate could be wide. It could be from 30 to 80%, and that needs to be communicated quite clearly. What information do we have or do we not have concerning protection against infection, mild, moderate, severe cases and deaths? And also that safety data will likely only be available for a relatively short period of follow-up time. The fourth point, or the third point, excuse me, is that some vaccines which are currently in development will have profiles which may provide important benefits to certain low and middle income contexts, meaning the number of doses delivered, the type of viral vector, the type of cold chain requirements, their suitability for wider use, their manufacturing capacity. And those things are an important point to consider when we're looking at potentially trialing these vaccines in low and middle income countries. The last 2 points are more about whether one should do a trial at all and how to do it and where. So one of the points there is to look at how do COVID vaccines fit into other priorities in the country itself. Is this the biggest problem facing the country? Is it not? Is it something that is important to them? There are very few vaccine trials in general and very few phase 3 trials which are conducted in low and middle income countries. So is this the priority? And that's last one is that both financial and career power relationships among those that may make the decision to fund or among those that may make the decision to do the trial certainly need to be taken into account. Those priorities may in fact be different than the priorities of the population itself. And that's it. Thanks, James.
Oh, thank you very much, Rebecca. There are quite a few questions, but I'll leave them for the end. So our last speaker today is Professor Gagandeep Khan from the Christian Medical College in Vellore, India. And Professor Khan conducts research on enteric infections and child health, and her team has evaluated vaccines in both preclinical and clinical phases for rotavirus and cholera. And she's a member of many WHO advisory committees, is the vice chair of the board of the Coalition of Epidemic Preparedness Innovations, chairs the Immunization Technical Advisory Group for WHO Southeast Asia region, and is also a steering committee member of this COVID-19 Clinical Research Coalition. So it's a great honor to have Professor Khan to speak at this webinar. Please go ahead.
Thank you, James, and many thanks to the coalition and the group for inviting me to speak here. Rebecca have done an outstanding job of laying out what the issues are with trial design and the complexities of actually doing trials in locations that need the product, but may not always necessarily need to be conducting the trials. So I'm going to be covering a lot of ground here, and I'll try to do it as quickly as possible. I think one thing that's important for all of us to understand is that for a vaccine to work in one part of the world does not mean that this vaccine is going to be immediately available in every part of the world or is going to be used in every part of the world. And to start off, I'd just like to give you an example of what product labels for vaccines look like and the kind of information that is available on those labels. These are the requirements or the formats that are used by 2 different regulators. And you can see that while the information is largely the same, the emphasis that is put onto different areas that are generated during the testing of the vaccines varies quite a lot. There is a different structure when vaccines are regulated separately from drugs, and in many countries, vaccines and drugs tend to be treated as equivalent to each other. So you can have a structure that looks at, for example, effects on the ability to use and drive machines, which most of us would not consider as being front and center when we think about the side effects of vaccines. So if we think about what regulators need to know in order to approve a vaccine, we start with preclinical testing, phase 1, phase 2, phase 3, and phase 4. And although quality relates to the manufacture of the vaccine, what I'd like to emphasize here is that from preclinical to all phases of clinical testing, including after licensure, safety is really paramount. We are measuring risk because vaccines are being given to healthy people. And it's only in phase 3 and in phase 4 that we look at efficacy pre-licensure and effectiveness post-licensure, which is what gives us information about the benefit of the vaccines. Now, if we think about regulators, we understand what regulators need. It is what sponsors provide to them as information on quality, safety, and efficacy. But if we think about what policymakers and the public require, then really it's a lot more. So if policymakers need to know that the disease is there and the disease has to be prioritized. This may be a public health priority, or it may even be a political priority, as we've seen in many low and middle income countries. There has to be alignment with global and with regional recommendations. There have to be estimates of disease burden and an understanding of whether vaccines are really the best way to control this disease or not. The performance of the vaccines, which is what comes through the regulatory system, is one aspect that is considered. But there are also economic and financial issues, the questions of availability that are not within the regulatory frame. And then there is the ability to actually deliver the vaccine, and that depends very much on the strength of the immunization program and the health system. For all of these, for decisions to be made, particularly in the context of a pandemic, we need robust and transparent data, a process for its evaluation, proven efficacy of the vaccine, and documented safety. Now, this is usually done through technical groups that analyze the information and present that to the government and to policymakers. But if we think about what is additionally required for the public, we really need credible countering of misinformation. And it's getting harder and harder. The kind of information that is being put out that is either appealing to emotions or quasi-scientific, at least in appearance, is getting more and more difficult to discount as a factor in how vaccines are accepted by the public. Now, I'd just like to take you through an example. The The Gamaleya Research Institute vaccine, which is actually two-dose vaccine that has recombinant AD26 and recombinant AD5 given as 2 doses 3 weeks apart, was licensed in Russia on the 11th of August 2020. We've had data from the phase 1 and phase 2 studies published recently. But this vaccine was licensed with a total of 38 volunteers ever receiving a dose of the vaccine. Of those, 18 received only 1 dose and 20 volunteers received both doses. The trial reported good binding and neutralizing antibodies and good cellular immune responses. And when we look at news reports, we were told that the clinical trials of the Gamaleya vaccine were over. But then after the outcry, the Russians have decided that vaccine trials on a much larger scale are needed. And for that, they have been in many countries, particularly in low and middle income countries. And I'd like to share with you what's been happening with the vaccine that's now known as Sputnik 5. The initial discussions with the Indian government started in August of 2020, and they had a tie-up with a company called Dr. Reddy's Laboratories, a large company, but not one that makes vaccines. Dr. Reddy's Laboratories requested permission in September for phase 3 trials. This went to the drug controller. They were denied the permission in October and requested to resubmit. They resubmitted for an adaptive phase 2-3 design, and this was approved last week. Essentially, what this consists of is 100 volunteers for phase 2 and 1,400 volunteers for what is called a phase 3 trial in India. Now, if we look at the Russian plans for a phase 3 study, that obviously is much larger. The goal there is to recruit up to 60,000 participants. And we've heard that they've recruited about 16,000 so far. And an interim analysis is planned for the 3rd week of November. Because Russia would like the vaccine used and tested in India, we've been told that Russia will send Indian regulators weekly updates from their trial. I'd just like to talk to you about what constitutes a phase 3 trial in the Indian context. For some reason, my slides are not moving. Okay, so if we look at what is a phase 3 trial, the example of typhoid in India, we started off with the typhoid vaccine that was a polysaccharide. This was called a phase 3 trial, but it only had 185 subjects, and it was the tip-bar vaccine was licensed. Then the company made a new improved product, which was a typhoid conjugated vaccine, again called a phase 3 trial, and with a total of 981 subjects, of whom less than 700 received the typhoid conjugate vaccine. Though the vaccine was licensed in India, it was not used in other parts of the world. So the vaccine was taken to Oxford, where it was tested in a controlled human infection model in a phase 2b trial, which showed that the vaccine had about 50% efficacy against the pre-specified outcome, but nearly 80% against a more clinically relevant outcome. When the vaccine was evaluated in Nepal, it actually showed also an approximately 80% efficacy. So what phase 3 meant in India and what phase 3 meant to other parts of the world were two very different things. I'm trying again to move my slides and it's not. So if we think about vaccines, this is complicated more by what constitutes full licensure, emergency use, and conditional approval. We know what an EUA is. It's when you don't have a full package, but because there is an emergency, you would like to be able to use this as a tool to control disease, but you would still need to continue enough data to go for a full licensure. But if you have an EUA or conditional approval from one national regulatory authority, what does that really mean for other countries? What does it mean for ongoing clinical trials? If you're using a placebo control, can you continue to do that when there is an EUA licensure? What does that mean when you define what the standard of care is? Can you use this for limited use, or can you take this now into a full public health immunization program? These are all questions that depend very much on the regulatory framework from each regulator. I'm sorry, I'm really having trouble moving my slides. So what are regulatory requirements if the vaccine is not manufactured in country? Essentially, it's two things. You can choose to trust other regulators, and that's regulatory reliance, or you may ask for bridging studies. And usually bridging studies are asked for if there are ethnic differences or environmental differences that might change the performance of a product. And these could be either only immunogenicity, or it could be a requirement for efficacy evaluation, again, depending on what the regulator would like to see from the package. Can we move to the next slide, please? So safety requirements during and after trials, I think this vaccine is going to be a little different. The clinical evaluation of safety, of course, is going to be there. But usually we have at least 6 months of safety data after completion of immunization, and this may be longer, a year or more for new technologies. In this case, the FDA has recommended 2 months and the WHO has recommended 3 months after participants have been vaccinated. And it's possible, given the rate of recruitment and the accrual of cases, that we may have an interim analysis that depends on case counts before safety data are available. So what will this mean in terms of the EUAs and then the subsequent follow-up of patients? In addition, there's also the question of laboratory evaluations, because we have concerns regarding vaccine enhanced disease depending on the platform. And there is a guidance that has been provided by FDA and other regulators that is asking for cellular immune response data. Usually this has not been a requirement in the more classical forms of vaccines. And there is a question about whether we should have requirements that differ depending on the kind of platform technology that's used. Can we move to the next slide, please? So efficacy evaluations, I think this has already been covered in terms of subgroups and the duration of protection. How long does protection last? Against what kind of disease? Against infection? Does the safety profile change with time, particularly with regard to reinfection? Do you get more severe disease? Does this work in subpopulations? And if so, are safety or efficacy different? The next slide, please. So just to end, the WHO Assure is a way of assessing regulatory authorities that ensures that we have some level of harmonization across national regulatory authorities. All national regulatory authorities need data, but it really is a question of having the norms and standards that we need, as well as good data that is generated and shared openly and transparently that will allow us to make the right decisions where vaccines are concerned. So thank you very much for your attention, and I'd be happy to take questions later.
Thank you very much, Professor Khan. Professor Kang. So there are a lot of questions, and I'll try and work through them. So the first question is for Dr. Grace. What are your thoughts on investing in testing candidate vaccines in low and middle income countries in parallel to high income countries, given the significantly lower burden of disease? And how generalizable would data from high income countries be in low and middle income countries, which is, I guess, the central question of this webinar.
Well, that's a heck of a question.
Yeah.
So the first one is, let me take a step back. So in terms of high income countries, depending on how you look at it, there's maybe 30 of those or so in the world out of all countries, one of which is the United States, which has one of the highest burdens of COVID. I think, I might be wrong, someone can correct me. And then if you sort of look at other locations where the burden of disease is higher, you do have places, certainly Cherry can speak to this, which is India, and then you also have Brazil. Those are not the lowest of the low in terms of low income countries, but they certainly are not considered high income countries. So I guess my first response to that would be is there certainly are trials which are being conducted today, or at least sites in larger multicentric trials, certainly in India, certainly in Brazil, in South Africa. And there is some discussion about potentially having a place like Kenya or other locations in Africa be part of multicentric trials. So that would be the first point. The second point is in terms of who's included in a vaccine trial and who the participants are is of course absolutely critical. If pregnant women, if elderly individuals, if those with comorbidities, are excluded from a vaccine trial, irrespective of where it occurs, that certainly has an implication in terms of the vaccine's generalizability in the future. I'm not sure I answered the question, but it's the best I can do right now.
That was a perfectly good answer for what is quite a yeah, big question. So the second question is from Philippe Guérin to all three speakers. The question is, could you explain what could be the product target profiles to address different epidemiological contexts, taking into account country incomes and capacities? And is it sufficiently debated by policymakers and vaccine manufacturers? So I don't know who wants to answer that question first.
I'm happy to take a stab at it. So I think when we think about low-income countries, low and middle-income countries, I think delivering a vaccine is something that needs to be prioritized much more. And if we look at both policymakers as well as funders of vaccines, the second wave that is being thought about is vaccines that are using manufacturing technologies that allow us to make a larger number of doses of vaccines that can be more easily delivered than the vaccines that are likely to be licensed first. So if you look at RNA and DNA vaccines, they are quicker to be made, but there are significant challenges in terms of temperature, cold chain capacity within countries that would make it very, very difficult for countries to be ready to take those vaccines on. Ultra high cold chain capacity is very limited in all parts of the world. If we look at different epidemiological contexts, I think here we could stratify on the basis of which vaccines work in subpopulations. So vaccines that work well in the elderly probably need to be reserved even more for high-income countries, whereas vaccines that work better in the immunocompromised for various reasons, either HIV or malnutrition, would need to be targeted to developing countries much more.
Rebecca Can or Rebecca Grace, would you like to add to that?
Yeah, so I just wanted to add on to what Cherry said. I think the first thing is it's important just to think about COVID vaccines as a continuum. And Rebecca Kahn mentioned this. There's what, 320 or 30 in development. And so the first generation, the ones that are the front runners, may or may not be the most appropriate for every context, which Cherry brought up. And it's much more likely to be that these second generation vaccines, or the ones coming second rather, are the ones that are most appropriate. And the criteria around those, as Cherry highlighted, might be slightly different. So one is it developed on a platform which is easily scalable into the billions? Is it something which does not necessarily entail cold chain requirements, which are quite so strict? Is it a vaccine where, if it's a vectored vaccine, is that using a vector which has been shown previously to be most appropriate for individuals who are immunocompromised? are there multiple doses required, which is likely to be the case, unfortunately. The route of administration, there's all sorts of criteria. And then a last one, which I would also add, in addition to what Cherry said, is also, is there a possibility of tech transfer, if you will? Is it possible? Is this a technology which could be transferred to a developing country manufacturer or to manufacturers who are aiming specifically to produce vaccines at scale? And that's something which is to be considered. I don't think that these things aren't being thought about, but it's perhaps not in as organized a way as one would wish.
And just to add one more thing, I think, following phase 3 trials and approval, there are phase 4 studies and other studies after vaccines are rolled out where we continue to learn more about their effectiveness and the other things that we didn't get from the phase 3 trials. So I think over time, we'll continue to learn more about all these vaccine candidates and see which ones work better in different settings. But I also agree the feasibility issues such as cold chain and number of doses are really important to consider.
Okay, great. So the next question is for you, Rebecca Khan, which is maybe a difficult one. Which of the current phase 3 trials is the best? I mean, the best design according to the criteria that you listed. I guess, have any of the current, the open protocols, or any of them mention subgroups as a stopping factor, or do any of them actually look at trying to estimate reduction in infection compared to just reduction in symptomatic disease?
Yeah, I think that's a good question. When I was reading through the different protocols, I was really struck by just how similar the designs of the vaccine trials really are, which I think speaks to the regulatory process. But they're really similar. Most of them are placebo controlled, or that they have an active vaccine. They pretty much all have the same primary endpoint of PCR positive with some sort of symptoms. The Oxford AstraZeneca trial does have a secondary endpoint looking at infection. And there are many other trials that have severe disease as a secondary outcome. I mean, if you look at these protocols, some of them have dozens of different secondary endpoints. So I don't think there's one that's better than another one necessarily. I think they're all very similar, and I think they're all thinking about all of these other questions. It's just, the primary endpoints are is an important endpoint and the same. But
I guess they didn't read your paper, maybe. They should have. So there's a question from Els Teril. So this is to both Rebecca's. Given that the trial design, including the definition of endpoints that are most relevant for assessing actual utility in curbing the pandemic are critical, how come this is left to companies and not imposed by public health authorities when they approve the trials?
So can I take that question actually?
Yes,
you can, Cherry. And afterwards, I was going to say, I'm happy to jump on after you. Go ahead.
So for low and middle income countries, between the regulators and the public health authorities, there's a divide, right? It's the regulators are supposed to be independent of what happens to public health. But I can give you an example of once where that did not happen. And the example is with the pneumococcal vaccine, where that was made by the Serum Institute of India that has recently been WHO pre-qualified. Now, most of the pneumococcal vaccines that have been made have been evaluated on a 3+0 immunization schedule. And because the package for Serum Institute was reviewed both by the regulatory authorities as well as by other arms of the government that are involved in policy making, there was an insistence that the vaccine had to be tested in a 2+1 schedule. So before the vaccine was licensed in India, it was actually evaluated on a 2+1 schedule and the data submitted to the regulators. That doesn't happen very often, but I think it is important. Over.
Rebecca Gray?
Yeah, I was going to try and respond to Elle's very poignant question, but one which is almost impossible to answer in 30 seconds. In terms of that, Else, I think my short answer to that would be that the development of vaccines, and in particular COVID vaccines, has been directed almost principally by large-scale manufacturers, and that isn't always the case. It's possible to conduct trials which are the sponsor or the groups of people who are deciding these things are not exactly the manufacturer itself, but are another entity. And in the case of COVID-19 vaccines, at least of the 7 or 8 front runners, that is the case. In terms of why there are not additional criteria imposed, if you will, by public health authorities or by other individuals, one component of that is what Cherry mentioned, which is regulators are supposed to be independent from this. And I think that what we've seen recently in the news is some discussion around how independent they are. And there's been some attempts to ensure their independence, and hopefully that will be the case in the future even more so. But also the fact that for lack of a better term, there's been some deficiency, let's say, in terms of leadership, if you will, in terms of the imposition of these criteria certainly among for the 7 frontrunners, which are from high-income countries or from powerful countries, if you will, in imposing that. And hopefully what we'll see in the future after this pandemic episode in the life of humans, that there will be some change there.
Okay, we're overrunning slightly, but I'm gonna ask one more question, which is actually from Professor Kang to Dr. Khan. Getting mixed up with the names, sorry. And the question is, with an inactivated vaccine or a codon de-optimized vaccine, nucleoprotein antibodies will not work, since the vaccines may induce them. And are there any other options other than frequent repeat samples?
Yeah, so that's a good question. So in the paper, we showed that if you can identify an antibody that can be used to distinguish between vaccine-derived immunity and immunity that comes from infection, then by sampling just a fraction of people once at the end of the trial, then you can get an accurate estimate of vaccine efficacy against infection. I think depending on the type of the vaccine, which antibody that is will differ, but if that can be found, then vaccine efficacy against infection can be estimated with fewer resources than we initially had thought, but it still does require additional resources to be testing not just symptomatic people. So it is an additional resource requirement, but I think we found methods that are lower resource requirements than we thought initially.
That's really, really important. Okay, well, we're a bit overrun. Thank you very much to the 3 speakers for giving these 3 fantastic talks. I'd also like to thank DNDi for putting this webinar together, and Julie Archer, who's the secretary of the coalition, and a lot of fantastic work. Thank you very much. Bye-bye.
Thank you.